In this section, we begin exploring one of the most fascinating systems of human biology, the reproductive system. Reproduction ensures the continuation of our species through the formation of gametes, fertilization, and development of new life. Throughout this chapter, we'll study how the male and female reproductive systems function individually and interact harmoniously to accomplish this goal. We'll look closely at the anatomy and physiology of reproductive organs, the hormonal regulation that drives the reproductive cycles, and how these processes lead to gametogenesis, fertilization, and embryonic development. This lecture presentation, prepared by Miriam Aladef-Feldman from Lake Washington Institute of Technology, provides a detailed yet clear overview of the physiological processes that underlie human reproduction. Let's begin by understanding the structure and function of the male reproductive system before progressing to the female reproductive cycle and developmental physiology. This chapter will guide you through 8 interconnected topics. We'll begin with sex determination, explaining how chromosomal signals decide whether a developing embryo becomes male or female. We'll then examine the basic patterns of reproduction, looking at gametogenesis in both sexes. From there, we'll move into the structure and function of male and female reproductive systems, followed by the processes of procreation, pregnancy, and childbirth, also known as parturition. Finally, we'll discuss growth and aging and compare homologous structures, the shared developmental origins of male and female anatomy. Together, these sections explain how reproduction is both a biological and developmental continuum. Humans are a sexually dimorphic species, meaning males and females are physically distinct. Reproduction requires the fusion of gametes, a male sperm and a female egg, to form a zygote, which develops into an embryo and later a fetus. This remarkable transformation depends on precise genetic and hormonal coordination, ensuring that development proceeds from a single cell into a fully formed human being. The gonads are the key reproductive organs, testes in males and ovaries in females. They perform 2 essential functions: producing gametes and secreting sex hormones. Males have XY chromosomes, while females have XX. Internally, both sexes begin with similar structures, called internal and external genitalia, which later differentiate based on genetic and hormonal cues. The presence or absence of specific genes determines whether these undifferentiated gonads become testes or ovaries. Every nucleated cell in the human body contains 46 chromosomes, or 23 pairs. Of these, 22 pairs are autosomes, and 1 pair consists of sex chromosomes. Females possess XX, and males XY. The eggs and sperm are haploid, meaning they carry only 1 set of chromosomes, 22 autosomes, and 1 sex chromosome. The sperm determines the sex of the offspring. An X-bearing sperm produces a female XX, while a Y-bearing sperm produces a male XY. Sometimes, abnormalities occur, such as Turner syndrome, where an individual has only 1 X chromosome, leading to unique physical traits and reproductive challenges. This image shows the human karyotype: 22 pairs of autosomes and 1 pair of sex chromosomes. Here, you can see that the X and Y chromosomes differ significantly in size and shape. While the X chromosome carries many genes essential for life, the Y chromosome carries a limited set of genes, including the one responsible for initiating male development. In this diagram, notice how X and Y chromosomes determine biological sex. Each egg cell produced by the female contains 1 X chromosome. Sperm, however, carry either an X or a Y. When a Y-bearing sperm fertilizes an egg, the resulting zygote develops into a male XY. If it's an X-bearing sperm, the offspring becomes female XX. Thus, it is the father's sperm that determines the genetic sex of the child. Early in embryonic life, Both male and female embryos have identical internal ducts, the Wolffian and Mullerian ducts. Around the 6th week, these ducts begin to differentiate under genetic control. In males, the SRY gene sex-determining region of the Y chromosome triggers testis formation. In females, the absence of the SRY gene allows the gonads to develop into ovaries. This marks the beginning of sexual differentiation. This figure demonstrates the bipotential stage of embryonic development at about 6 weeks. At this time, the reproductive organs have the potential to develop into either male or female structures. If the SRY gene is present, testosterone and anti-Mullerian hormone AMH promote the development of male ducts and the regression of female structures. Without these signals, the Mullerian ducts form the fallopian tubes in uterus, and the gonadal cortex differentiates into ovaries. This figure beautifully illustrates how the same embryonic tissue can give rise to two very different reproductive systems. In humans, reproduction follows a sexual pattern involving gametes from two parents. Female gametes, or oocytes, are large, nutrient-rich, and non-motile, while male sperm cells are small and motile, equipped with a flagellum to swim. Females are born with all their oocytes, which mature one per month from puberty until menopause. Males, however, continuously produce sperm from puberty throughout life. This difference defines the contrasting reproductive strategies of the sexes. This figure compares the process of gamete formation in males and females, known as gametogenesis. On the left, you see oogenesis, the production of female gametes or eggs. It begins before birth, when oogonia undergo mitosis to form primary oocytes, each containing 46 chromosomes, or 4N DNA. These primary oocytes enter meiosis I but pause in prophase until puberty. During each monthly cycle after puberty, a few primary oocytes resume division. One completes meiosis I, forming a secondary oocyte and a small first polar body that soon disintegrates. The secondary oocyte, now with 23 duplicated chromosomes 2N, is released during ovulation. If a sperm cell penetrates the secondary oocyte, it triggers meiosis II, producing a mature ovum and a second polar body. The nuclei of the sperm and ovum then fuse to form a zygote, restoring the diploid number to 46 chromosomes. Thus, one primary oocyte ultimately yields just one egg. On the right side is spermatogenesis, the formation of male gametes. Here, spermatogonia divide by mitosis throughout life to produce primary spermatocytes, each with 46 duplicated chromosomes. Through meiosis I, these primary spermatocytes divide into secondary spermatocytes, and then through meiosis II into spermatids, each containing 23 chromosomes. Finally, spermatids mature into spermatozoa, or sperm cells. Each primary spermatocyte produces 4 functional sperm, in contrast to the single egg produced from one oocyte. Together, these processes ensure that gametes carry half the genetic material necessary for the creation of a new individual, restoring diploidy at fertilization. Reproduction is orchestrated by the brain through the hypothalamic pituitary gonadal axis. The hypothalamus secretes gonadotropin-releasing hormone GnRH, which stimulates the anterior pituitary to release follicle-stimulating hormone FSH and luteinizing hormone LH. FSH regulates gametogenesis within the gonads, while LH controls the production of steroid hormones such as testosterone, estrogen, and progesterone. An enzyme called aromatase converts androgens into estrogens, maintaining hormonal balance. This complex signaling system ensures synchronization between the brain and reproductive organs. Both sexes produce androgens and estrogens, but in different proportions. In males, testosterone, produced mainly by the testes, is the dominant hormone responsible for male characteristics, libido, and sperm production. In females, estrogens, including estradiol and estrone, along with progestins, dominate the hormonal profile. These hormones regulate ovulation, maintain the uterine lining, and support pregnancy. Additionally, the adrenal glands in both sexes secrete small amounts of sex steroids, contributing to body hair distribution and libido. Understanding this balance of hormone production is essential to grasp how the brain coordinates the timing and function of the reproductive system. This diagram summarizes the hypothalamic pituitary gonadal axis, the central command system of reproduction. At the top, the hypothalamus releases GnRH in rhythmic pulses. Those pulses travel to the anterior pituitary, stimulating it to secrete FSH and LH into the bloodstream. From there, these gonadotropins reach the gonads, the testes or ovaries, triggering them to produce sex steroids such as testosterone, estrogen, and progesterone. These steroid hormones then circulate back to the hypothalamus and pituitary to create a feedback loop that keeps hormone levels within an ideal range. The figure also reminds us that this axis is not a one-way path. It's a continuous dialogue between brain and body, adjusting reproductive activity in response to age, stress, and environment. Reproductive hormones operate through feedback inhibition and stimulation. Normally, increasing levels of testosterone or estrogen signal the hypothalamus and pituitary to reduce GnRH, FSH, and LH release, maintaining balance. However, there's a special exception in females. When estrogen rises rapidly for more than 36 hours, it actually stimulates the pituitary instead of inhibiting it, producing the LH surge that triggers ovulation. The gonads also secrete peptide hormones that fine-tune this system. Inhibin suppresses FSH, while activin enhances its release. This intricate web of signals ensures that egg and sperm production occur in perfect rhythm with hormonal cycles, aligning fertility with physiology. Finally, we turn to the neural timing of reproduction. Deep within the hypothalamus lies the GnRH pulse generator, a biological clock that regulates the frequency of hormone release. These pulses are crucial for the onset of puberty, the maintenance of fertility, and the rhythmic nature of the menstrual cycle. External factors can influence this system. Stress, nutritional status, and even daylight exposure can alter hormone rhythms, sometimes delaying puberty or disrupting menstrual cycles. Modern research also points to environmental estrogens, chemicals that mimic natural hormones, as potential disruptors of normal reproductive function. Together, these insights highlight the delicate interplay between the endocrine system and the environment, reminding us that reproduction depends not only on anatomy but on a finely tuned network of brain signals and external cues.